Hi, everyone. Welcome back from break. We're going to get started with our next session, which is exploring the landscape of gene therapy. For those of you who may not know me, I'm Eric Camino. I'm PPMD's Vice President of Research and Clinical Innovation. Before we jump into this session, I just want to give a little bit of housekeeping on how we're tackling some of these sessions this year. Over the different conferences, we get a lot of feedback from families, and what we've heard is that at times, some of these talks where we get into research and investigational products and approved products, it can be a lot of information kind of take on at once in these sessions, and so we wanted to try something a little bit different this year, where we'll continue to have those talks, but rather than have a number of our industry partners on stage, we're going to have some of our clinicians and researchers present all of this material. So you'll notice as we go through the conference over the next few days, when we get to these sessions, we won't have any industry partners on, just our clinicians and researchers from the field. Now, we still wanted to make sure that you had an opportunity to have that kind of direct connection with our industry sponsors. So on the CVent app, you can schedule one-on-one meetings and connect with companies throughout the meeting. And then on Saturday, after we've covered all of the investigational products, all of the approved products, we're going to have a 2-hour block called the Pharma Forum, where over in the exhibit hall, we'll just have kind of a 2-hour period where you can go around, you can talk to these companies, have some more discussion, have some more questions answered. But we also hope that during these sessions, we're able to answer a lot of your questions. And so our focus for this session is to talk about gene therapy, some background education information on this for families who this may be newer to, some information on kind of the challenges of understanding kind of the immune response, and then we'll go of an overview of the investigational products that are currently in development, and then we'll have an open Q&A with all of you. So to get started on kind of that background, the history of gene therapy, where we're at, where we're going, we're going to be joined by Dr. Jeffrey Chamberlain from the University of Washington to kind of provide that overview.
Thank you, Eric. It's nice to be able to be here today and give you a little bit of background on gene therapy. So this slide has my disclosures on here. So I just want to start off very basic and hopefully go into a little more detail as we move along. First of all, I mean, what is gene therapy per se? How do you define gene therapy? It's the simple concept is to use genes as a form of medicine, a type of a drug. The idea behind gene therapy is to deliver a new gene to the body or to directly manipulate the structure or the function of a defective gene. And there's a couple of different types of gene therapy out there. Gene replacement therapy is probably the most common version. That's where you just want to deliver a new gene into the body, regardless of what's already happening there. There's a second approach called gene knockdown, where you actually need to try to shut down or inactivate a gene that's causing problems. It has inherent toxicity of its own. And one of the newer methods that probably a lot of people have heard about is gene editing, which is a method to try to directly go in and to modify or to repair a gene to fix the mutation, and that uses the method known as CRISPR. So, the inherited diseases are caused by mutations in different genes, and they can lead to either inherited or sometimes acquired diseases. And these inherited or so-called genetic diseases often run in families, but they don't always run in families. There's a number of diseases, particularly Duchenne muscular dystrophy, where there is a high rate of new mutation. In other words, nobody else in the family has a mutation in the gene, but an individual that's born has acquired a spontaneous new mutation that leads to the disease. There are more than 6,000 different inherited or genetic diseases that are known. Two of the most common ones are of course, Duchenne muscular dystrophy and cystic fibrosis. Genes themselves, most genes make proteins. Not all of them do, but most of the ones that are involved in a lot of the common inherited diseases are genes that produce a protein, and the proteins are what do the actual work inside of a cell. And there's the two main types of mutations that are found in different types of muscular dystrophy. There are so-called dominant mutations, and if you have a dominant mutation, what that means is that the gene is still producing something that is damaging the cell. On the other hand, you have the other class of mutations are known as recessive mutations. And in a recessive mutation, you have a gene that basically doesn't do anything. It doesn't produce a protein, or if it does, it doesn't produce a protein that's harmful in any way. And that's certainly the case in Duchenne muscular dystrophy. It's a recessive disease. Most of the mutations that lead to Duchenne muscular dystrophy simply inactivate the gene. It doesn't do anything. And that's what makes it a candidate for gene replacement therapy to try to bring in a new copy of the dystrophin gene. Now, there's obviously the related form of Duchenne muscular dystrophy known as Becker muscular dystrophy. And the difference between Becker and Duchenne is that in Becker muscular dystrophy, you have mutations in the gene that lead to either low levels of the so-called dystrophin protein that comes off that gene, or you're producing a protein that's only partially functional. And the result of that is you have a slightly milder version of the disease that's slowly progressive. And so the idea of trying to apply gene therapy to these recessively inherited diseases is to take a new version of the gene, usually a synthetic or laboratory manufactured version of the gene, and try to get it back into the muscles of the body to take over and replace the function of the defective or the mutant gene. And so what I've drawn at the bottom of this slide here is a little cartoon of a typical gene, and this is how we put together the genes that are usually delivered in the context of a gene therapy trial. Often you're not just taking the normal version of the gene and trying to deliver it. Often it's too big. It doesn't necessarily lend itself to being easy to deliver. So typically what you do is you take the part of the gene that makes the protein that you're trying to replace. And then you have to attach onto that an on-off switch that basically turns the gene on in the muscle cells and keeps it off in other cells. Those are known as promoters, and I'm going to come back to that in a minute. So you have to put together a choice of a promoter to turn the gene on and off. You have to have the part of the gene that makes the protein you want to deliver, and then there's also a very small signal at the end of the gene which tells the cellular machinery that it's reached the end of the gene and it can stop. So a little bit of the timeline of the development of gene therapy for Duchenne muscular dystrophy. This is an ongoing timeline. Unfortunately, we're not really quite there yet. But just to give you a history of what's been involved and how long work in this area has been undergoing. The field was really set in motion back in around 1986 by the incredible work primarily of Lou Kunkel at Harvard Medical School, who identified the dystrophin gene, which is the defective gene in Duchenne muscular dystrophy. And some people point to that discovery as the beginning of the Human Genome Project. It was the first time that someone was able to go in and identify a disease-causing gene without knowing anything about the gene itself. And Ron Wharton up in Toronto also played an important role in identifying the dystrophin gene. Soon after that, Kay Davies over at Oxford in England published an incredibly important study where she identified a patient that had an extremely mild form of Becker muscular dystrophy, yet she showed that this patient was only making a small version of dystrophin. In fact, the mutation of that patient took out half of the entire gene. And normally, if you take out that much of a gene, you're going to completely inactivate it, yet this individual was able to make reasonable amounts of this really small protein and had an incredibly mild version of the gene. And that set in motion a lot of ideas on how one could potentially develop gene therapy, and I'll come back to that a little more in a little bit too. Myself, I got involved in the field around that time, and we were able to publish the first study where we could take a dystrophin gene and find a way to introduce it back in the muscle cells. Later in the 90s, a couple of laboratories, Barry Byrne, Jim Smolsky, and Jim Wilson, published a series of papers suggesting that this delivery vehicle known as AAV was actually pretty good at getting in the muscle cells, and it suggested the possibility of using these AAV delivery shuttles as a way to do gene therapy for different muscle diseases. The problem with these AAV vectors is they're really small, and the dystrophin gene is really big, and the two didn't go together very well. But work that had been going on in my laboratory for quite a while was able to identify really small versions of dystrophin that had a reasonable amount of function to them. We named those microdystrophins, and we showed that these microdystrophins could in fact be carried into muscles using AAV, and that led to another study in my laboratory where we showed that if you could prepare these AAV vectors carrying these microdystrophins at really high quantities and inject them directly into the bloodstream, that they would actually exit the bloodstream and deliver their cargo or their dystrophin gene to muscles throughout the body. And that really set up for the first time a way that one could potentially do gene therapy for muscle diseases. Now, it was many years before that was able to be translated into clinical trials. The first clinical trials of muscular dystrophy were done by Jerry Mandel's lab in Columbus, Ohio, at Nationwide Children's Hospital. And eventually, just in the last year or so, Sarepta was able to take some of these early AAV microdystrophin delivery vehicles and get them approved by the FDA for treatment of Duchenne muscular dystrophy. So the timeline there, it's 37 years between the identification of the Duchenne muscular dystrophy gene and the first FDA-approved drug. So that's a really long time. And the question is, where do we go from here? And the one thing I will say is that even though it took so long to get here, so many of the tools that were developed over the years, not only in the studies of muscular dystrophy, but many other different diseases, have really accelerated the field, and progress is coming much faster than it used to. So what are the challenges really of trying to develop a Duchenne muscular dystrophy gene therapy? And this also applies to Becker muscular dystrophy. So there are really three key components that I think everybody in the field, including myself, has focused on for many years. One is the gene itself. What is it that you want to deliver back to muscles in order to try to treat this disease? Now, obviously you would like to deliver the dystrophin gene, but that's very difficult to do. The dystrophin gene turns out to be the largest gene known in any organism, and it's about 200 times bigger than a typical gene. So nobody's really figured out a way to manipulate the entire dystrophin gene, much less deliver it into the muscle, and that's what's led to the idea of making these miniaturized versions of the gene, these so-called microdystrophins or mini-dystrophins. The other thing, as I alluded to earlier, with the gene being so big, we can only use a small portion of it, so we have to add back onto it these so-called on-off switches, these promoters. And so you want to figure out a way to come up with the ideal promoter that will make your protein, your dystrophin protein, only in muscle, but not in other tissues. You particularly don't want to make it in liver or in immune cells. It could activate an immune response and fight off the gene therapy. And then finally, the vectors. What is a vector? A vector is a way to try to deliver genes to a target cell. You can think of a vector as sort of like a shuttle bus to transport people from one region to another. So a vector is a way to take a synthetic gene and transport it into the body and drop off its payload, the dystrophin gene, into the muscles of the body. So when work in this area began more than 30 years ago, we really didn't know the answer to any of these questions, and so a lot of the research over the years has gone into what is the best way to develop a gene that could be delivered, how can you keep it active in muscle but inactive in other cells, and how are you going to even get it into muscles all over the body? The human body is almost 40% muscle. It's an enormous target area to try to deliver a new gene to. And so those are the main challenges being faced in the field. So again, and some of those challenges, particularly as they apply to Duchenne and Becker muscular dystrophy, are dealing with the dystrophin gene. I mentioned it's by far the largest known gene in nature, but the good news is the defective gene is silent. It's not causing any active damage by itself. The problems you get in the muscles of patients with Duchenne is that they're not working properly because they're missing a protein, but the defective gene is not in and of itself causing problems. And again, this gets us back to the idea of working with smaller dystrophins. They're a lot easier to manipulate in the laboratory. And the idea of using these miniaturized dystrophins, so-called mini-genes, has also become important in alternate therapies such as exon skipping and even in the CRISPR gene editing strategies that are starting to show some more promise. excuse me, at the bottom of this slide is a little cartoon of of what we know about the structure of the dystrophin protein itself. The upper cartoon there just shows the different parts of dystrophin. It's got a lot of different pieces that do different things inside muscle cells. And below that is the original mini dystrophin that I referred to earlier that was identified by Kay Davies back in the late 80s. That was the first mini dystrophin suggesting that you didn't have to deliver the entire gene back into the body. So over the years, my laboratory and a couple of others kind of worked on different ways to try to engineer the dystrophin gene to figure out, one, what are the most important parts of this gene? Since we know that Becker patients are often missing parts of the gene, what really are the parts that you have to keep in there? What are the parts that are dispensable? We started with that mild Becker patient. We tweaked it a little bit and showed we could improve its function by cutting off actually some pieces that were causing some problems. And then beyond that, we started removing a whole bunch of different pieces of dystrophin, testing them out one at a time, and identifying what are the parts we need to keep in there, what are the parts that we can throw away and still get a pretty functional protein. And that led to the development of what's shown at the bottom is a couple of different versions of these so-called microdystrophins. And again, the advantage, why would you want to deal with a microdystrophin? Why don't you just deliver the whole gene? Well, what I would say is that if we could, we would deliver the whole gene, and I'll show you some data later on that we may actually be close to being able to do that. But in the meantime, right now, we can't really do that. The only delivery shuttle that is known that can efficiently deliver new genes to muscles throughout the body are these AAV vectors, and these AAVs are so small they can only hold about 1/3 of the dystrophin protein, and that requires that you deliver microdystrophins rather than larger dystrophins. And I should point out that the work and the development of these microdystrophins was done by a brilliant graduate student, Scott Harper, who worked in my lab about 20 years ago. So the other question I mentioned, you need on-off switches. How do you regulate a gene in muscle so that it only turns on in muscle and is off in other studies? And a lot of this work has come out of a close colleague of mine, Steve Hauschka at the University of Washington. Steve's had a long-standing interest in how muscle genes are turned on and off during development, and he focused in on this gene called MCK. And Steve and I were able to show many years ago that the MCK gene is completely silent in all non-muscle cells, but when muscle begins to form in the developing human embryo, this MCK gene is switched on and it comes on to very, very high levels. And so Steve has gone on and developed hundreds of different promoter on-off switches from the MCK gene, and several of these are being very widely used in gene therapy trials across the world. So then the final issue, how do you deliver these dystrophin genes to muscle? You know, I mentioned that back in '97 was the first demonstration that these so-called AAV vectors could work really well in muscle, but they were only being delivered by doing an injection right into a single site of muscle, and it didn't spread. So, you know, you've got this small shuttle, it doesn't hold large genes. We were able to fit these microdystrophins into it, but we still had this idea, how are you going to deliver it all over the body? And that was a solution that we started working on about 25 years ago, and the solution to that was come up with by a postdoc in my lab named Paul Grigorovich, who's now down at the University of Melbourne in Australia. Paul did some experiments of injecting increasingly larger amounts of these AAV shuttles directly into the bloodstream, and even with the delivery of enormous amounts of the AAV into the bloodstream, nothing was getting into muscle. And then we got to this breakthrough where we figured out a way to make even more amounts of AAV, and once we hit a certain critical threshold event, suddenly these vectors started leaking out of the bloodstream and into the muscles. And the example I'm showing you here is from a mouse model of Duchenne muscular dystrophy that normally doesn't make any dystrophin, and after a single injection right into the bloodstream of these mice, we were able to deliver these microdystrophins to every muscle of the body. And this is really what's paved the way for all the muscle gene therapy trials ongoing throughout the world today. So AAV, though, turns out not to be a perfect vehicle for delivery. The dystrophin levels that we're getting, even at the highest safe doses, are a little lower and more uneven than what you would really like to have, certainly lower than what we see in the mice. They're able to slow disease progression, but they can only partially reverse a loss of weakness. Higher doses would probably overcome that, but that's not safe. We're already really at the upper limit of safety here. So what are some ways to do that? One is to come up with more potent AAV vectors that will work at lower doses, and there's been a lot of progress in that area in the last couple of years, and some of these newer, more potent vectors are starting to come into the clinic. Gene editing is one possible solution. It's not quite really ready for prime time, in my opinion, but it'll be there someday. We need stronger promoters. We need to control the immune system. A lot of the problems historically with gene therapy have been because of immune system activation that causes adverse events you want. And then ideally, you'd like to be able to deliver larger dystrophins. The microdystrophins are only 1/3 the size of the full protein. They're only partially functional. Are there ways to deliver larger proteins? And so that's one of the things that we're working on now in my laboratory very hard, is a way to deliver larger and more functional dystrophins. And the way that we've come up to do that uses co-delivery of either 2 or 3 of these different AAV vectors. And what we found is that we could take different pieces of the dystrophin protein, put them into completely separate AAVs, co-deliver them, and by putting a little tag on the ends of these 2 proteins, they can spontaneously come together and make larger proteins. And by co-delivering 3 AAV vectors, we have a paper that's in press right now, shows that you can make very, very high levels of the full-length dystrophin by co-delivering these AAV vectors. So here's an example of that. Again, this is a cartoon of the dystrophin protein itself, which is what you want to produce. At the top is the entire dystrophin protein, showing in the different colors a number of its different pieces and parts that are important for its function. At the middle is what we call a mini-dystrophin that we can now produce by co-delivery of 2 different AAV vectors. And at the bottom is one of the microdystrophins that's in the clinic that can be delivered with a single AAV vector. So the current therapies use 1 vector to make microdystrophin. I think we're on the verge of having ways to use 2 vectors to make a mini-dystrophin and 3 vectors to produce the entire dystrophin protein. So this is just an example, again, from mouse studies where we've developed this. On the left is an example of a relatively low dose of AAV delivery into the mice to make a microdystrophin. And with a conventional AAV vector, such as AAV9, that particular dose doesn't really produce any of the protein that you need in muscle. But if you switch over to one of these new classes of these muscle-specific or myotropic AAVs at that same low dose, you can produce very high levels of this microdystrophin protein. Now, what's been exciting to us is if we go to the 2-vector system where we're bringing in 2 different pieces of dystrophin, we can deliver those back into the mouse at the exact same dose that you use to deliver the microdystrophin, and we're able to make large amounts of this mini dystrophin protein. And on the right, it's a little harder to see. Basically what I'm showing there is this is an experiment where we co-delivered 3 different AAVs into a dystrophic mouse, and when we extract the dystrophin out of the muscles of that mouse, all of the muscles throughout the body of those mice are now making high levels of the full-length intact dystrophin, and we've been able to show that the full dystrophin is significantly more functional than the mini dystrophin, which is significantly more functional than the microdystrophin. So, unfortunately, this is still experimental in the laboratory, but we hope over the next few years to try to translate this approach into the clinic. So finally, to close out a little bit, what are really some of the prospects for gene therapy of Duchenne Becker muscular dystrophy moving forward? It's not a perfect therapy. It's better than anything we've had up till now. I think it is. I would say the major limitations, though, are the doses. The very high doses that we need to deliver even as much microdystrophin as we can are somewhat limiting. They can lead to some very serious adverse events, as we've heard. Even at those high doses, we're not making quite enough dystrophin, and these microdystrophins are probably not as functional as we'd like them to be. There's also pre-existing damage to muscles such that some of the older patients are maybe a little harder to treat than the younger patients. But we feel that if we can co-deliver 2 or 3 different AAVs, using the new myo version or these new class of AAV vectors, that we might be able to deliver larger and more potent dystrophins at lower doses than what are currently being used in the clinic. Now, there's certainly a couple of other things under development that are coming along that I'm not going to go into, but there's a lot of options out there that are being developed that need to be developed. So I'm going to just stop right there. Thank you very much for listening.
Thank you, Jeff. And as Jeff was talking through that, one of the challenges he did bring up was that, when we get with delivering AAV, we do have kind of these immune responses. So we have Dr. Craig McDonald from University of California, Davis to talk through some of what that looks like with current gene therapy.
Great, thank you very much, Eric. So I think this is really an important and certainly a timely topic, the immune response in AAV gene therapy. These are my disclosures. Today, this afternoon, we're going to be discussing both the innate and adaptive immune responses to gene therapy. We'll discuss the time course of immune-mediated reactions and adverse events with gene therapy. I'm going to touch upon some of the preventive treatment protocols that are currently used in AAV gene therapy, some of the current approaches to the treatment of serious adverse events, and then I'll close out by discussing really some of the differences of these two recent acute liver failure events leading to death in 2 patients and the future approaches to potentially mitigate risks. Just to start with, in terms of the mechanisms of gene therapy toxicity, it's really our natural immune system and our immune system's ability to fight off infections that really arises or gives rise to the problems of the immune responses with giving a viral vector or AAV gene therapy. And these really can be discussed in terms of the very rapid onset innate immune responses and the issues with pre-existing neutralizing antibodies, which can inactivate a vector and make it really not functional, but also could potentially produce some safety risks. Those innate rapid onset immune responses can give rise to complement activation, which ultimately can give some very early and serious side effects, low platelet counts or thrombocytopenia. In some of the earlier programs, a process called TMA or thrombotic microangiopathy, or a variant affecting the kidneys, atypical hemolytic uremic syndrome. And then there's an adaptive response, and these immune responses can occur either to the viral vector or to the, or also referred to as the capsid, or to the replacement gene, the transgene itself. So there can be adaptive responses to the capsid, adaptive responses to the transgene, and ultimately we can see issues such as myocarditis, myositis, or an inflammatory myopathy, a reaction to the transgene, or also the most common side effect, which is a liver issue or hepatotoxicity. This is data from the Sarepta programs, the observed timeline of the capsid versus transgene-related adverse events in clinical trials with the Elevidys or delandistrogene moxeparvovec. And so there are specific adverse events specific to the transgene product and also those that are driven more by the capsid. But some of the earlier adverse events relate to complement activation, thrombocytopenia, early cardiac events, liver events, which occur typically from 4 to 8 weeks, and then rare cases of immune-mediated myositis or muscle events. And these later events are T-cell mediated and typically occur from 4 to 8 weeks. And this is actually data-driven. This is some of the data from the Sarepta clinical trials. The earlier dot there in each line is the median time of onset of these various adverse events. And then the dot on the far right is the median duration of the adverse events. So generally, the event of myocarditis is something that occurs within days of the gene therapy and oftentimes can take place with a duration for the first months. So again, the innate immunity is our first line of defense. These can be things such as physical barriers, epithelial cells, soluble factors, non-hepatopoietic cells, immune cells, cells called neutrophils, basophils, eosinophils. The exposure here leads to an immediate maximal response. It's non-specific, and there's no memory involved here in terms of the immunity. And then so-called adaptive immunity, which is really our second line of defense, which involves B cells, T cells, antibodies. There's usually a lag time between exposure and maximal response, and it's pathogen antigen specific, and it also is we see long-term immunologic memory occurring. So in terms of the initial timing of the immunologic responses to AAV gene therapy, you can see down at the bottom, you have decreasing systemic vector concentration in the bloodstream that occurs largely over the first month of AAV gene therapy. And with this initial innate immunity, we see complement activation, issues of antibodies, neutralizing antibodies, macrophages, neutrophils. And then later on, usually occurring in the time frame of say 4 to 8 weeks and can extend beyond, is really the acquired immunity, which is causing problems with things like liver toxicity, as well as the myositis. But this is generally, most of these are generally T cell activated responses. And there have been a number of prevention approaches that have actually treated these so these are the standard peri-infusion steroid regimens. Sarepta, for instance, uses prednisone 1 per kilo for generally for about 3 months, starting 1 day prior or 1 week prior to a steroid-naive patient. The Solid Biosciences program uses a higher dose of steroids for the first month and then tapers to 1 per kilo. And the REGENXBIO program, for example, uses prednisone. There have also been emerging other additional immune suppression regimens that have been utilized. For instance, the REGENXBIO group is using eculizumab to try to prevent complement activation side effects, and then they're also using sirolimus as well. So in terms of the antibody-mediated immunologic responses to AAV capsids, there's really this potential for pre-existing innate immunologic responses to a vector. Some of the neutralizing antibodies can initially block the transduction of the vector, making it non-efficacious. And then there's also non-neutralizing antibodies, which also, along with the neutralizing antibodies, can be involved in complement activation and also presentation of the antigen to macrophages as well. In terms of this initial early response of complement activation, this largely produces early onset thrombocytopenia in really quite a predictable manner. Usually the low point for platelet counts is usually within the first 6 to 8 days of infusion with complement activation. Fortunately, with the Elevidys program, there have not been reports of thrombotic microangiopathy, or TMA, or kidney end organ involvement, hemolytic uremic syndrome in that program. But in some of the previous gene therapy programs, we did see some of these TMA and atypical hemolytic uremic syndrome events. Here's an example of data from the Sarepta program showing a reduction in some of these complement factors. They're cleaved to other components in the complement cascade, but at about 1 week, you see this reduction in some of these complement factors, and with this, you see this reduction in platelet count occurring at about 1 week after gene therapy. Unfortunately, we've not seen in the currently FDA-approved product, we haven't seen these more severe complement-mediated events, such as thrombotic microangiopathy. This is characterized by microvascular thrombosis, endothelial injury, presents as hemolytic anemia, consumptive platelet count reductions or thrombocytopenia, and end organ damage. And in addition, there have not been cases of hemolytic uremic syndrome where you actually get acute kidney failure. One of the immunosuppressants that I mentioned that's currently being used by REGENXBIO is eculizumab. This actually binds to the complement protein C5, inhibits cleavage to the C5a and C5b components, and really is effective at mitigating this serious complement activation that we see. Another event that we see early on after gene therapy is heart-related events or viral myocarditis, which can be due to a viral involvement of the myocardium itself or an immune response in the heart, affecting the heart muscle to the viral vector. In myocarditis with AAV gene therapy, typically the time course again is days to a month. This is an inflammatory disease of the myocardium. Again, it can be toxic, infectious, immunologic. Acute myocarditis is usually a fairly short onset of symptoms, presentation usually early on, sometime in the first month, but it can be occurring in the first days. It's characterized by elevated blood troponins, wall motion abnormalities by imaging studies, and evidence of inflammatory infiltrates in the myocardium. And usually patients will present with these elevated troponin I levels, decreased cardiac ejection fraction by echo or MRI, focal cardiac wall motion abnormalities, T2 signal abnormalities, and changes in cardiac MRI as well. The incidence is fairly low, 1%, but we really need to point out that most of the patients in the Sarepta clinical programs had left ventricular ejection fractions that were higher, greater than 48%. The 40% criteria was an inclusion criteria, but very few patients in those trials had these low cardiac ejection fractions, and I think we have to keep that in mind when recommending AAV gene therapy to somebody with impaired heart function. And then moving over to some of the muscle events, the immune-mediated myositis, again, is T-cell activation. It's due to T-cell activation. It usually occurs from 4 to 8 weeks in terms of timing. This, again, is a T-cell mediated immune reaction to certain characteristics or certain epitopes of the replacement gene, of the transgene, which causes an inflammatory myositis diffusely in multiple muscles. This has been related to mutations involving exons 8 and 9. They're currently excluded. There's caution with regard to exons 1 through 17 and 59 through 71. Patients present with severe limb weakness, increasing proximal weakness, upper limb weakness. They can also have weakness of bulbar muscles affecting speech and swallow, weakness of respiratory muscles leading to transient ventilatory support. Fortunately, this has been mitigated by excluding patients with mutations of exons 8 and 9 with earlier mutations which involve that particular epitope. But we should recognize that there have actually been probable cases of immune-mediated myositis which have emerged beyond exons 8 and 9 in the global experience that Roche has been involved with. And the treatment thus far has been IV methylprednisolone, in one case tacrolimus, sirolimus, and plasmapheresis as well. And then we can get also issues with regard to liver toxicity. And I think that's really the most serious and consequential adverse event is this later onset liver event due to T-cell activation. With regard to acute liver and AAV gene therapy, generally these patients, about 30 to 35% of patients will actually have elevation in AST, ALT, GGT. These are the definitions that Sarepta uses for acute liver injury, a GGT elevation as well as AST elevations and alpha elevations. There's also a definition for acute liver failure, which involves elevations in bilirubin greater than two times the upper limits of normal, as well as INR elevations. But it also includes development of ascites or encephalopathy as well. And there have been patients that have had these elevated INRs, which is a synthetic factor involved in coagulation. These patients, though, have not gone on to develop ascites or encephalopathy, so they've not been characterized as liver failure. When generally the GGT is increased to three times the upper limits of normal, or a level of, say, greater than 100 to 200, prednisone, oral prednisone, the peri-infusion steroid is increased, usually to 2 milligrams per kilo. And with further increases or an increase in the bilirubin or INR, usually IV methylprednisolone is used. And second-line agents with worsening acute liver injury is generally IVIG, 2 grams per kilogram over 2 days, or sirolimus has been utilized as well. Again, methylprednisolone is typically used oftentimes either 1 to 3 days, typically doses of 30 milligrams per kilogram. The IVIG has also been used. Dr. Veerapandiyan has a report of series of cases where he has also successfully added IVIG to the methylprednisolone regimen. And then more recently, sirolimus has been used. This actually inhibits mTOR signaling and actually dampens that T cell activation. And this is the medication that Sarepta is considering as in their new immunosuppressive regimen that a number of experts will be trying to develop in the coming weeks. I do want to just make a couple of comments here about these 2 recent deaths. These have been in non-ambulatory patients, 15 and a 16-year-old. They presented at 4 and 7 weeks post-treatment and died within 3 to 4 weeks. And the acute liver failure was really characterized by severe increases in the liver function test, the transaminases, well beyond what we typically see with acute liver injury. But also what was really unique about these cases is severe cholestasis. They had significant elevations in their bilirubin values beyond a level of 10, elevations in their alk-phos, and they also had impairment in synthetic function. So one of the coagulation studies called an INR actually was quite elevated in these patients, and they also developed low serum albumin, which is also indicative of low synthetic action. And in one case, these 2 cases developed severe coagulopathy. In one case, death occurred due to an intracranial hemorrhage in the first case. But also what has been unique about these cases has been late onset thrombocytopenia with platelet counts as low as 20,000 in a very late occurrence after, say, 8 weeks after treatment. And in one case, low platelets were responsive to ramiplostim or N-plate with the platelet count coming up nicely, and platelet transfusions were used in the other case. But aggressive late immunosuppression after the onset of acute liver failure with sirolimus and multiple other agents were unsuccessful in the second case. I think there's an interesting question about this thrombocytopenia seen with acute liver failure. I think it's obviously under investigation by Sarepta, but I think we should keep in mind that thrombocytopenia is a condition that's actually commonly seen associated with acute liver failure. This could be due to various factors: reduced thrombopoietin production, which is a hormone that stimulates platelet production, increased platelet consumption, and potential autoimmune destruction of platelets as well. But thrombocytopenia in acute liver failure seems to be a risk factor for the development of multi-organ system failure and relatively poor prognosis. I want to just close by mentioning there seem to be some similarities with the experience with X-linked myotubular myopathy, AAV gene therapy deaths. There were 4 deaths, unfortunately, in the X-linked myotubular myopathy program, and this seemed to be correlated with hepatobiliary dysfunction, which was part of the underlying condition, the X-linked myotubular myopathy patients. Many of them have evidence of hepatobiliary dysfunction before the gene therapy. And these cases also had cholestatic liver failure with really high, very high bilirubin levels. We know now that, I think many are asking, why is ELEVIDYS on hold for non-ambulatory patients? What is it about non-ambulatory status? And I think really non-ambulant status is really a gross proxy for disease severity. I think are some concerns for metabolic changes and underlying pre-treatment hepatobiliary dysfunction from steroid treatment, insulin resistance, and emerging data concerning the presence of non-alcoholic fatty liver disease or steatosis in Duchenne patients. And there's a recent publication that came out that examined a large number of Duchenne patients with a variety of tests, including ultrasound measures. A high proportion of Duchenne patients had significant fatty liver or hepatic steatosis. The ultrasound Nagami parameter index actually seemed to increase with disease progression, with DMD progression, and fully 2/3 of the non-ambulatory patients with Duchenne dystrophy had moderate to severe fatty liver changes in their liver. And so we know that fatty liver disease, fat accumulation can lead to inflammation, fibrosis, causes the liver to become stiffer and so forth. And we've shown that transition to ambulation actually is associated with increases in BMI and weight gain. This is literature we published. But just to close, in terms of future approaches, ELEVIDYS is on voluntary hold by Sarepta for the non-ambulatory population for both the ENVISION trial and commercial patients. Sarepta will provide an ELEVIDYS pre-treatment and regimen for 12 weeks with sirolimus, which will be developed by an expert academic panel. Some clinicians may opt to actually treat both ambulatory and non-ambulatory patients with sirolimus in the future. And I guess one of the questions is, can we identify better risk assessment for the development of acute liver injury or acute liver failure? Eculizumab is being used for the first month. That's not Insmed, but REGENXBIO is using eculizumab. And I think future approaches, which Dr. Flanigan will discuss, is viral vectors with greater muscle tropism, allowing lower dose of AAV, really detargeting the liver. And then also, which is really quite exciting, intrathecal delivery with lower dose AAV microdystrophin gene therapy with the Insmed program. And with that, I'll stop and move over to Dr. Flanigan. Thank you.
Thank you, Craig. And then our final talk before we move to the open Q&A is going to be from Dr. Kevin Flanigan from Nationwide Children's Hospital, who's going to kind of walk us through the investigational products. So these are the ones that are currently in clinical trial.
Thank you very much. In the old days of vaudeville, they used to say, never follow dog acts or magicians because you could never live up to them. And it's tough to follow Dr. Chamberlain, Dr. McDonald, because of the excitement and energy. And that's what I meant, the fascination, not because anything bad. So I'll try to keep you as engaged with that. I'm going to talk today about 4 different programs. Typically, one begins with a disclosure slide, and I apologize, I don't have a disclosure slide. I am involved in doing trials with several of these companies, so I'll make that clear, but I'm here representing just the field, not any single company. I want to make that clear. I have no horse in the race, so to speak. So I'll begin with Insmed. So Insmed, you heard about just a minute ago, is taking a unique approach to gene delivery, to vector delivery. It's a one-time intrathecal injection of virus, so into the spinal fluid by lumbar puncture. It uses a fixed dose. It's not a weight-based dose, but really a fixed dose. And the idea is that it winds up distributing ultimately to muscle and to heart. So it's a microdystrophin gene, similar to ones you've seen before. The study is called ASCEND, a single-dose injection of this. Preclinical studies carried out in non-human primates have shown that this leads to efficient transduction and expression of microdystrophin in muscle. That data has not yet been published, and I'm hopeful that it will be published in time for as enrollment starts for the study, so parents can evaluate that themselves. But the preclinical data does look really compelling in this regard. With this advantage to this, it might allow much lower doses of virus, leading to lower risk of systemic reactions. This holds up in animals. This first-in-human study is to show whether that's true in patients. So ASCEND is the first-in-human study. It's a phase 1 multi-center study to look at the safety. There's no placebo group. Enrollment is for children ages 2 to less than 5 years old, ambulatory boys with a definitive DMD diagnosis, the single injection overseen by an independent data monitoring committee. Participants will be followed for 100 weeks post-treatment with an extension study to monitor safety for an additional 3 years. I mentioned the CLIA inclusion criteria, the diagnosis of DMD and age. Patients need to be able to walk without support for at least 10 meters and to cooperate with and perform motor assessments. Exclusionary criteria will be prior treatment with gene or cell-based therapy or oligonucleotide-based exon skipping. And patients will be excluded based on the presence of deletions encompassing the exons 1 through 17 or 59 through 71, and of course the typical antibody, AAV antibody screening as well. Boys will be also excluded if they have signs or symptoms of cardiomyopathy, which would not be expected in this age group. This is where the study is being performed at multiple sites across the country. There's a QR code there. I know folks from Insmed here are at the meeting as well if you'd like to talk to anyone about it. Really, the primary outcome is to evaluate the safety and efficacy of this approach. And the key secondary efficacy outcome is to look at expression in muscle. So for questions, at the bottom you have the people to reach, medical information at insmed.com if you're interested in enrolling in that. The second ongoing trial is REGENXBIO. You've heard some about already. REGENXBIO, these are their forward-looking statements, not mine, but I'll just flash through those quickly. This is a microdystrophin AAV gene therapy, a phase 1, 2, 3 clinical trial called Affinity. I think you guys are very familiar with this, but I'll just point out the components of this are an AAV9 vector, the promoter, the on-off switch Dr. Chamberlain mentioned, a different promoter than has been used in previous trials, SPC512, and a microdystrophin transgene that differs from some of the other transgenes, and I'll show you how that is. I'm going to call your attention to two different ones. I'm going to make use, we're going to come to SOLIDS trial in a moment, so I'll point out both of those vectors, and we can compare them in some ways to the microdystrophin vector used of Sarepta. I'm afraid the pointer won't work very well. Oops, I'm going to go back one. Pointer, it does. On the left side here, I'll show the pointer. Each of these shows the N-terminal actin binding domain of the protein, some number of spectrin-like repeats, the C-terminal dystroglycan binding domain that binds to the membrane. And what's distinct about the REGENXBIO is the way these have been shuffled. They include a portion of the gene after the dystroglycan binding domain. This portion of the gene binds other proteins that are they're binding sites for proteins like dystrobrevin and syntrophin. Whether these are important or not in therapeutic replacement of the gene will be determined by a study like this, but that's one of the ways that shows it's distinct. Just because I have this image up, I'm going to share with you, we're going to come to SOLID in a few minutes. SOLID has a different shuffling of the spectrin-like repeats, and this is a binding region for another protein that I'll mention in a minute. But this is what's distinct about REGENX's transgene, the transgene itself. So the trial, it's for boys aged 1 to less than 12 years old with genetic confirmation of DMD, no pre-existing antibodies. Really, there's two groups of patients there, 1 to less than 4 years old that can do 10-meter walk without assistance, that are on a stable dose of corticosteroids, and those 4 years and above, which the only difference is the younger group has to be above 10 kilograms. As mentioned earlier by Dr. McDonald, one of the features of this trial is the use of a proactive immune modulatory regimen. This includes eculizumab, which inhibits complement activation and theoretically at least would have an impact on the innate rapid immune response that Dr. McDonald mentioned. and then as well as sirolimus, which was a T-cell directed a drug, often used in transplantation and other therapies as well. In addition, there's elevation of prednisone during the course of the trial. So the idea here is that this may potentially reduce the sort of safety signals that were described. So for the phase 1/2 trial, again, safety is the primary endpoint. Biomarker endpoint is microdystrophin expression with a variety of secondary endpoints of muscle function. Those of you who've been to many of our clinics or been involved with trials will recognize the time to stand, the 10-meter walk-run, the time to climb 4 stairs, the North Star Ambulatory Assessment, and so forth. I'm going to show some data that provided by REGENX, looking at 2 dose levels for this. Dose level 1 on the left, dose level 2 on the right. Most of the data that I'm going to show you is the dose level 2 on the right, consisting of just 3 boys. I'm sorry, I can't read it from here. I think it's 3 boys from ages 1 to 3, and 7 boys. I'm sorry, now I'm getting old. In my old days, I could read that stuff, no problem. And you can see that these, some of the demographics, they're quite similar between the groups. You can see starting North Star Ambulatory Assessment, for example, at 24, around 24 and so forth. I'm going to skip ahead and show some of the important data regarding safety. So far, there's been no safety signal in all 13 of these boys treated with this. No safety signal marked by central or peripheral neurotoxicity, which is of concern. There have been some questions whether the capsid used, AAV9, might be in some ways neurotoxic. There have been no signals of drug-induced liver injury, thrombocytopenia, myocarditis, or myositis that's been mentioned before. So this has been well tolerated in this. So this is looking at the biomarker of microdystrophin, and what you see is a couple of different ways that we measure microdystrophin. In the second row, that's microdystrophin by Western blot, a method where we can look at and quantify the amount of dystrophin in a muscle fiber, in a muscle sample. And at the bottom is percent positive fibers, another number that many of you will be familiar with. And we can see at dose level 2, we have levels of, in the younger patients, of about 120% dystrophin by Western blot, ranging to 54 in the middle group, and around 39.7 in the older group. These are small numbers of boys, obviously, but these are what the numbers are. And percent dystrophin positive fibers similarly range from 82.1 to 45.7. There is significant tropism of muscle. This measure here, vector copy number, is a way we measure how many copies of the virus itself gets into the tissue of interest. So the vector copy number is expressed here as copies per nucleus. So for every nucleus in the muscle fiber, it ranges from almost 25 to 18 copy numbers per nucleus. It's a kind of a surrogate measure of tropism of muscle. There's some limited clinical data on this so far at 9 months. In the next slide, I'll show you at 12 months. But it does suggest, compared to external controls, improvements in the North Star Ambulatory Assessment in the 5 patients, in the higher dose group patients. Only 5 of these are included here, with North Star Ambulatory Assessment increased by 4 points at the end of 9 months, but a trend in all the measures, time to stand and so forth, to show improvements compared to external age-matched controls. And similar numbers hold up at 12 months as well. So somewhat promising data. So to summarize, the biomarker data really suggests robust expression. This seems well tolerated, and the functional data support consistent evidence of this microdystrophin, this approach, altering the trajectory of disease in comparison to natural history controls. The company is moving on with a pivotal trial, a phase 3 pivotal trial. Again, it's very similar criteria, as you can see. Really, in this case, with adding the North Star Ambulatory Assessment criterion of a greater than equal to 16 for boys who are ambulant 4 years and above. The same regimen, same immune modulating regimen with eculizumab, sirolimus, and increased prednisone, and very similar timed endpoints. The primary endpoint in this pivotal trial would be the proportion of patients with greater than 10% microdystrophin levels. And this is being conducted at multiple levels across the country. Again, I would note to you there are representatives of REGENX here if you're interested in discussing anyone in that group. So, and here's another number, another email address if you're interested in reaching out to anyone about that trial. Third trial is Solids, SGT-003 trial. This is another variant of microdystrophin therapy with two major variations associated with it. I pointed out earlier to you on the slide on REGENX the difference in the organization, the shuffling, if you will, of the components of the dystrophin gene. The two repeats I pointed out are the binding region for nitric oxide synthase. Nitric oxide synthase is it's a protein that its presence seems to improve blood flow to prevent activity-induced ischemia and presumably reduce associated muscle injury. So that's one thing that's different. And the second thing that's different is the capsid. This is a different viral envelope, an engineered version of the viral envelope. It's called SBL101. Derived, most closely related to AAV9, but with some engineered changes to it. And it has increased tropism of muscle in preclinical studies. Tropism is the term we mean, does it go to those tissues? So increased muscle tropism and relative decreased liver tropism for it. So this combination is now used in the trial, first in human, in which that's being done. So this is the INSPIRE trial. The INSPIRE trial is a single dose level, open label, phase 1-2 study for patients with a diagnosis of Duchenne. It's actively enrolling in the United States, in Canada, and in Italy. There's a regulatory study level approval in the UK, so another site is expected to open there soon. It uses prophylactic prednisone as its immunosuppression. And there's anticipated expansion into cohorts, additional cohorts to increase older and non-ambulatory boys. The primary endpoints is safety, this incidence of treatment emergent adverse events through the 1-year time point, and change from baseline of microdystrophin levels at day 90, with a variety of secondary endpoints that you'll recognize: time to rise, 10-meter walk, run, and so forth. There are 3 cohorts, ages 4 to 7 years old, ages 7 to less than 12 years old, and a new cohort that is just starting to enroll of ages 0 to less than 4 years old. Patients are excluded if they have deletions of exons within 1 through 11 inclusive, or within exons 42 to 45, because this microdystrophin encodes those repeats encoded with 42 to 45. So there are no functional requirements for the youngest and oldest boys for enrollment in cohort 2. I'm sorry, the youngest and middle boys for enrollment in cohort 2, the ages 7 to 12 years old. There are criteria for time to rise and 10-meter walk run. Once again, the eligibility criteria require being negative for antibodies to AAV9, no history of gene therapy, and a washout period from things like exon skipping therapies, vamorolone or givinostat. One has to be on prednisone for this during this therapy. So the steroid regimen on cohorts 1 and 2 is a stable dose of daily oral steroids, prednisone or deflazacort, for the 12 weeks prior to enrollment. Those are switched to prednisone during the period of the trial. And there's no steroid regimen required in that youngest cohort we've added. I'm going to show you data the company released after their data lock in March from the first 7 patients, only 3 of whom had their biopsies analyzed by that time. So the 3 patients will show the day 90 biopsy and biomarker data. You can see the age range here from 5 to 8 years. The first thing we'll note is evidence of that there were no treatment emergent serious adverse events, and all treatment-related adverse events resolve without any sort of sequelae in the weeks following dosing. So there's specifically no evidence for hepatotoxicity, with no biomarker or clinical evidence of liver injury, no GGT results. You can look at the graph at the bottom, the figure at the bottom, no GGT level elevations with this. And none of these patients required eculizumab or sirolimus. The 3 patients that were biopsied have shown some dystrophin. These are dystrophin expression studies from that. The first thing I'll note, again, that vector copy number measure with a median of just about 19, or a mean of about just about 19 vector copies per nucleus, suggesting significant tropism, significant viral entry into muscle. Microdystrophin protein as expressed in muscle is shown here two different ways: Western blotting that I mentioned before, and another quantitative measure called mass spectrometry. The Western blotting itself shows levels of about 110%. The mass spectrometry matches that with levels of about 108% of normal. And again, this measure of dystrophin-positive fibers as measured by interval fluorescence shows close to 80% of fibers that are positive. On the left is dystrophin staining, kind of recapitulating the last slide. In the center column here, marked in green, is staining for something called beta-sarcoglycan. Dystrophin re-expression, in the absence of dystrophin, the complex of proteins to which it binds is altered. So when we re-express dystrophin by any means, we look for the re-expression of these members of the complex. So beta-sarcoglycan is our surrogate measure for the whole complex, restoring that complex. And what we see here is comparable to the dystrophin expression. We can see that there's significant re-expression of beta-sarcoglycan. On the right is nNOS activity, measured by a different method, by an immunohistochemical rather than an immunofluorescent approach. And this also shows significant restoration of nitric oxide synthase activity at the muscle membrane, as one would expect based on the structure of the microdystrophin. I won't show all the data, but there's significant increases in markers of muscle integrity observed at day 90 in these first three participants, including serum CK, serum transaminases AST and ALT, other measures such as serum titin, which is a measure of muscle damage and a potential surrogate biomarker in some trials. Cardiac biomarkers, left ventricular ejection fraction, I'll point out in 3 boys. These are small numbers of patients. None of them showed a decline in left ventricular ejection fraction. One patient was enrolled with an elevated troponin, slightly elevated troponin, that increased over time at 90 days. We can't take too much information from these small numbers of patients regarding the heart, but there's no signal suggesting toxicity. So that's it. Just to summarize, there's really quite a few vector genomes per nucleus, clear microdystrophin expression, both by Western blot at 110% and percent dystrophin-positive fibers at 78%, with restoration of binding partners that are important. Consistent improvements across 7 biomarkers and no serious adverse events. This trial is still enrolling. There are certainly representatives from SOLID here as well, and I would encourage anyone to contact those individuals if you're interested in finding out more about this trial. And I'll close with Sardocor, which is a somewhat different approach. Here we're not talking about delivering microdystrophin gene therapy. This is involving a cardiac gene therapy, direct delivery by coronary injection, coronary artery injection, into muscle. Sardocor has a delivery of a gene called SERCA2a. SERCA2a is important in function of calcium handling in muscle and in heart. It's been tested, its overexpression of SERCA2a has been tested in other forms of adult heart failure, with safety fairly well established in those settings, in more than 120 patients. The objective here is to assess safety and biologic activity and cardiac function in the setting of Duchenne dystrophy. So SERCA2, the protein SERCA2, is essential for what's called contraction and relaxation that occurs via coupling to calcium, so calcium reuptake in these tissues. SERCA2a is naturally occurring in heart cells, but is known to be decreased in people with heart failure and with Duchenne dystrophy. And increasing SERCA2a has reversed heart failure characteristics in multiple preclinical models. That's dog, pig, and mouse, if you can't see that from the back. The study is called MUSIC. It's a phase 1b open-label multi-center trial that will enroll up to 12 adult males. 6 will receive this gene therapy, SERCA2a. 6 will be observational controls. It's intracoronary artery delivery at 2 dose levels, so it requires an overnight hospital stay. There'll be 2 years of active follow-up with cardiac MRI and biomarkers and functional assessments, requiring 10 study visits over 2 years with annual visits thereafter. The primary outcome is safety with all-cause mortality as the defined primary outcome, adverse events related to the treatment, immune responses to SRD-001. Exploratory outcomes are efficacy, including the characteristics of heart muscle by cardiac MRI, cardiac biomarkers, including troponin and BNP. There will be a variety of measures that test upper limb function, the pull test, grip, pinch, and elbow flexion test, and pulmonary function test, and the DMD quality of life questionnaire. So an overview again of this, you have to be a male above 18 years old with Duchenne, left ventricular ejection fraction of less than 50%, and the presence of myocardial fibrosis on cardiac MRI, corticosteroid use for at least a year, stable on an optimized regimen for your cardiac care, capable of pulmonary function testing and capable of doing cardiac MRI, and exclusionary for patients who have a pacemaker or an implantable cardiac defibrillator, who have severe lung impairment with forced vital capacities of less than 25%, or have had prior gene therapy or have liver impairment. So there are a couple unique aspects to this. These are adults with Duchenne. These are people, I don't have to tell this audience, often excluded from other clinical trials, looking for patients with cardiomyopathy with low ejection fraction, non-ambulatory adults, independent of what the mutation is, and one can take other therapies during the trial. And even if one is antibody positive, you can participate in the trial as part of the control group. There's data sharing on the cardiac data, which is helpful, so that it doesn't have to be repeated. A cardiac MRI with gadolinium does not have to be repeated for an individual. And direct administration to the heart means there's lower total doses of delivery of AAV. Dr. McDonald's great talk, one of the take-homes of this is that all of these immune responses clearly seem to be related to at some level to the total AAV dose that's delivered, not just the per weight dose. So by giving relatively low doses in an injection like this, it's a relatively low amount, even though it's a non-ambulant and older and bigger boys. So I'll end here to say that there are 3 clinical trial sites led by real experts in the Duchenne cardiac space, Dr. Mammon at the University of Kansas, Dr. Villa at Cincinnati Children's Hospital, and my colleague Dr. Nandi at Nationwide Children's Hospital. So with that, I'm happy to close and move on to our next portion. Thank you.
All right, so I'm going to invite all of our speakers back up, actually, so we can move into the open Q&A, as well as Dr. Tim Kripe from Nationwide Children's Hospital, who doesn't treat Duchenne patients, but is an expert in gene therapy and on PPMD's board as we kind of move into this discussion. Also, while the speakers are coming back on stage, if you're looking for more information on any of these clinical trials, you do have in your bag a booklet that goes through and lists out kind of all of the ongoing recruiting clinical trials, and then we do have more information on PPMD's website with our Explore Clinical Trial page, which we just recently updated. So we will open up to questions. So we have mics in the middle of the aisles. So if you have any questions, please line up, and then we will start to go through those. I think I did what, yeah.
Yeah, I actually have a commentary to start with and a question for the other esteemed members. It was a terrific overview, guys. Really appreciate that. My commentary is I think people should understand we're really at the end of the beginning, not the beginning of the end. I mean, there's so much more. It should have been obvious, I guess, with all the investigations that are still ongoing, that there's still a lot to learn. And when it comes to the approvals and so forth, the trials have been relatively small numbers of patients. So we're not going to identify rare signals or rare events in small numbers of patients. So as we get more and more real-world experience, I mean, if you look at the cancer trials that I'm more familiar with, we'll often have 1,000 patients in them. So, as we get more and more real-world experience, it's natural for things to sort of come out that are rare. One of my questions for the panel is, we know from other areas of AAV gene therapy that a lot of the immunogenicity can come from the structure of the DNA. DNA is foreign. There are these, anytime you have a C next to a G, it's known to be a toll-like receptor, the unmethylated CpGs, as it's called. It's been identified in the hemophilia gene therapy world as very important. And so now the vectors are being made to reduce those numbers of immunogenic CpGs, if not eliminate them altogether, not only in the coding sequence, but also in promoters and regulatory elements. Are any of the vectors that you guys described from these different companies reduced purposely for CpGs and therefore might be less immunogenic?
I can't speak for all the companies. I'm not aware that people have done that so far. There's certainly been a lot of codon optimization for other reasons to try to enhance how much protein is made from these vectors after they're delivered. But this is kind of a new and emerging field of study that is starting to suggest that this could be an important way to try to reduce the immune reactions against these vectors. I mean, particularly Bradley Hamilton at Stanford has done a lot of work on this, and he's still trying to gather data on which changes are the most important ones to make. It's a little hard to predict exactly where you want to reduce these, C's and G's in the vector. But I think that's going to be an emerging area of study where people are going to have to start changing the sequence of the DNA in their vectors. But are you guys aware of anyone that's already introduced that?
I'm not aware of that, other than codon optimization, which is a different sort of story.
Right. I think it's just an illustration of how much more work there is to do and learn and make these better.
And then I did want to ask, or comment as well on one kind of idea is we saw a number of years ago, so in the talks that happened, we talked about kind of exon 8 and 9 being excluded both in some of the labeling for something like a Levitus, but often also those clinical trials will kind of see those exclusions. And that was something that we started to see in some trials. They talked about the immune-mediated myositis, so instances where the protein that's being produced kind of reads as foreign to your body, and so your immune system can start to attack your cells. So these patients are excluded. We saw a really great effort where the companies and members of NIH came together to say, we're seeing this, and sharing data to try and limit this risk for patients, and we currently see these exclusions. So with all of these different vectors that are being used, and we're talking about different, with different tropisms and different serotypes, how well can we kind of look across all of these different Duchenne trials that are all using AAV, but sometimes different doses, and kind of take shared learning so we can get to a place where we have better safety. We also saw in this that there's a lot of different approaches for the immune suppression drugs that are being used. So I think for families, probably there's a little bit of like, what appears to be right? What's the best choice for me? And that's, it's hard to kind of wade through all of this.
I'm happy to take a stab at that. So, that observation of exon 8 and 9, for example, really came out of open sharing among the companies at that time, Pfizer and Solid and Sarepta, Genethon. That was driven actually by academics, to be honest with you. It was our colleagues Francesco Montoni and Carsten Boneman who, I don't know that they twisted arms, but they made a compelling case that was in the interest of all the companies to be fairly open about these events that occurred. They put together a couple of workshops, and it led to the New England Journal paper that really describes across multiple platforms. I think the challenge now is how do we ensure that the companies that are doing each of these studies maintain that sort of openness with it. Certainly, I don't mean to cast, companies don't want to lose competitive advantages of this, but it's clear that it moves the field forward by people being fairly open about these events, sharing what's the genotypes of these events, sharing the treatment trial. So that New England Journal coming out of that first workshop of sharing, I think set the bar for what we have to do moving forward.
Thank you. And then we'll start over here and then come to the middle.
Hello, I am Dana Edwards. Hi, Dr. Flanigan. See you September 23rd. And Dr. Chamberlain, I don't know if you remember me. I started writing you back in 2008, and I wrote you every Christmas to make miracles happen. Happy New Year. Yeah, Happy New Year, Merry Christmas, and save my son. I wrote you year after year. I just looked them up, and you were telling me that you were starting the gene therapy, and you were hopeful. Anyway, Tanner's missing 3 through 36, so he doesn't qualify for anything, and he was asked to participate in the Pfizer study, And thank God he opted to say no because he's over 18. And when they asked him, and the young man passed away in that study. So when Sarepta, I was hopeful. And I want to see this do well. I want something for my son because he does have a rare mutation. And it didn't work out. I have a question. It might sound stupid, I don't know, but have you ever thought about combining gene therapy and stem cell to mitigate the immune response? Stem cells are really like an amazing something for our kids. I mean, immunity and all those, keeping them well, their heart, their lungs, it's really doing well. And I don't know if you ever thought about a combination of that, maybe because it's not FDA approved, I don't know.
Yeah, if I could just comment, there is a cell therapy that is currently been in open label extension and placebo-controlled trials. That's Capricor's Duramiocel, which actually utilizes cardiosphere-derived cells. There's really no, the data on the open label extension using objective MRI data really looks spectacular, and there's an FDA AdCom considering approval that'll take place in late July. There's really no reason why a therapy like that could not be used synergistically with gene therapy. It wouldn't necessarily be done at the same time, but certainly sequentially, a therapy like that, a cell therapy like that, it doesn't, these cell therapies oftentimes don't work through engraftment. They really work where the cells actually hone to, say, the pulmonary vasculature, and they secrete these vesicles or membrane-laden vesicles called exosomes, which actually have the cargo, which actually are helpful to the disease process. So they don't, the cell therapy or the stem cell therapy don't really work through engraftment, but they have systemic effects. And originally, that therapy was actually done with direct coronary artery infusions, similar to the last gene therapy approach with the SERCA2A that you heard about. But then we actually moved into IV administration, which I think was a lot less burdensome to patients and turned out to be seemingly efficacious and safe.
Could I add one thing to it as well? So, and just because I want to be clear for this community and parents, you know, when we talk about stem cells, you know, there are these places you fly to Dominican Republic, you give them a bag of cash, and they'll say they'll inject you with stem cells.
Yeah, I'm not saying do that.
Yeah, don't do that. That's fraudulent. There's no evidence of those things. But there's really interesting work. Again, I have no skin, no relationship with the company, but there's really also interesting work specific to the stem cell population of muscle, which are called satellite cells. And clever approaches to those tire out over time. Satellite cells in muscle, when your muscle degenerates, these cells make new muscle and then repopulate themselves. And over time, when muscle is being continuously turned over, like in Duchenne dystrophy, those peter out. And a colleague of ours, a really great scientist, Mike Rudnicki, has developed an approach that really looks like, in animal models, an orally available agent that re-stimulates those satellite cells in a way that may in fact be the first sort of muscle stem cell directed therapy to reach clinical trials. And I'm not touting it, it's only animal data, but I do know it is reaching clinical trials within the next calendar year or so. So we may exactly be in that position where we're giving a gene therapy or an exon skipping therapy or whatever, and at the same time reboosting the stem cell population that lives in muscle already.
Okay, thank you. Oh, I'm sorry.
I was just going to say the idea of combination therapies, I think, is the next frontier, but it's really hard to test two unknowns at the same time. And like you implied, you know, not FDA approved yet. So I think that's going to be the future, what you can combine together and how best to use them.
Yeah, no, thank you.
And then we'll come here in the middle.
Oh, great, thank you. I have a 16-year-old ambulatory son with DMD, and gene therapy is available to us right now unexpectedly, still fairly, frequently, recently. And what does it look like to have a maximum dose of the viral vector and doubling his already maximum dose of steroids for 3 months. Like, I don't have a picture of that. Do some of you all have experience with that? And what are we looking at if we actually decide to go forward with this?
So, I'm sorry, I'm just, so I'm going to start by asking first question. So anytime I hear about a 16-year-old boy with Duchenne is still ambulant, in my book, the most common of the patients I see, and I'm enriched a little bit because we study this under sort of NIH grants and things, but usually those boys are making some degree of dystrophin for some reason. Their mutation is somehow permissive to make some amount of dystrophin. So in boys who are diagnosed with DMD at a younger age, but really stand out apart from the standard course of DMD, I always suggest that before parents consider going down the irreversible and one-time therapy with gene therapy, to really consider doing a muscle biopsy to determine, do you have one of these permissive mutations that allows you to make some dystrophin? Now, I'm sometimes asked by parents how much means I should take it and how much I shouldn't take it. That's not the answer we get. The answer is the reason your son doing well because he's making some dystrophin his own, and how do you factor that into the risk-benefit ratio of all the risks you mentioned? Mm-hmm. Or if your son is completely absent of dystrophin, then you can't attribute what he looks like today to that benefit, and then you would say, well, some amount of dystrophin from these therapies might be worth the risk. So I acknowledge all of the risk that you just said, but the further a young man looks from what's typical Duchenne, and I will say it depends very much on the mutation as well. We can look at the mutations and say, is this potentially permissive about this? I will say I'm not, I probably take this approach more frequently than many people. You might not hear this from everybody, but I know I'm not alone in it, and I see Stan standing over there, so I'm hoping I think there's many people who would agree that this is a reasonable approach. We can do a needle biopsy, determine what it is, how much is there, and weigh that into the factoring of risk-benefit of what you might get out of it.
And for other families who are looking at this, who are at the same sort of max dose already, do you all have experience to say how intense is that? And thank you so much for what you said. I just don't want to waste everyone's time with my specific.
Yeah, I've certainly, as a general question, I have dosed some patients, both in the trials and commercially, that have been, up in the 80 kilogram weight range. The maximum dose is really, with the commercially available gene therapy, is sort of maxed out at sort of a 70 kilogram equivalence. I think some of it really depends on the specific situation. I think if you had a larger child right now and who is ambulatory, I might just pause and wait and see what the experts come up with the sirolimus planned immunosuppression protocol, because I do think some of the larger ambulatory patients, potentially could, benefit from some of that immunosuppression. I've had patients who were larger that have done well. I've had one patient who was larger that actually developed acute liver injury, and certainly going, the steroid dose at the 1 per kilo range usually peaks out at 60 milligrams a day, and with acute liver injury, when you double that to 120 milligrams a day, it's pretty challenging for patients to be treated on a daily basis with that high a steroid dose. The patient ended up that I had with 80 kilos ended up doing well, but I do think that's a pause for concern, when sort of approaching the larger weight patients, in this current era. Fortunately, we have additional approaches that hopefully can lower the dose that may make acute liver injury less less problematic, and I think some of the immunosuppression is going to be really helpful as well.
Thank you.
Stan?
Stan?
Yeah, hi. I agree with you, Kevin, whatever you say, of course. So my question's a numbers question. It's probably directed at you, Jeff. You've been doing this for 30 years. And for some of the data that Kevin was showing in terms of the current vectors in therapy now dosed at 1 to 2 times 10 to the 14th viral particles per kilo, we're getting delivery of 20, 30, 40 vector genomes per nucleus, which seems like that battle is then won, and maybe even a lower dose could have been effective, and you sort of talk about a threshold effect. So keep that in mind. Then the second part of this question is Steve Hauschka's data of how wildly more effective these promoters are than, say, for instance, the naturally occurring dystrophin promoter, the naturally occurring CK promoter, which is far more active than dystrophin. All of these should be expressed at much higher levels. So I've got kind of a little bit of a numbers problem that I just love your thoughts about in terms of, are we getting 20 viral vectors in? Are they there, but only a tiny portion or even possible to transcribe? Are some of them being turned off transcriptionally, leading to the fact that we get to maybe near normal amounts of protein at the end of it? Thoughts?
Real quick, just to hop in before then, and I do want your answer, but just can we re-explain vector genomes for maybe families who are kind of hearing this and they're used to hearing either just like dystrophin expression or just so they kind of, again, the concept of what we're talking about when we say vector genome copies.
Okay. So yeah, I mean, I think the issue is, you know, how you have to deliver this AAV vector and get it into muscle, and then it's turned on and it starts making this dystrophin protein. So when people talk about vector genomes per nucleus or per unit body weight, they're really just trying to quantify how much of the AAV is accumulating in each part of the muscle cell, in all the different muscle cells. And what Stan is asking, which is I think is a fascinating question. This is actually something we've been talking about quite a lot in my own laboratory lately. There seems to be some contradiction in what's going on here. You know, as you mentioned, the artificial promoters that are being put into these AAV vectors we know they're so strong, they should be just blasting out microdystrophin and making far more than normal levels of dystrophin. On top of that, we're seeing many copies of these AAV vectors getting in the muscle to the point where you would predict you should be making 100 times more than normal dystrophin. And yet, you know, we've seen these companies, and in the best case scenario, they're getting up to about normal levels of dystrophin. And that's not a lot, because dystrophin is actually a very rare protein. It's one of the harder proteins to find in muscle. It's not normally made at high levels whatsoever. So the data suggests you should be making hundreds of times of normal dystrophin, but you're not. And why is that? Unfortunately, the simple answer is we don't know. But something is clearly not happening the way we would predict it to. And I think the theories that are out there is that, you know, when you're trying to quantify how much AAV has gotten into the muscle, typically you're just taking a whole chunk of muscle, you're grinding it up, and you're asking how much AAV was in this chunk of muscle that I took out in a biopsy. The problem is that doesn't tell you where in the muscle it is. Some of these vectors, some of these AAVs might actually be outside of the muscle cell and they never really got in, but they're stuck there and they're still present when you grind up the muscle. Others, my guess is others are getting into the muscle, but they're not going to the right place in the muscle so that they can be turned on. In other words, they're not making it into the nucleus. These AAV vectors have to not only enter a muscle cell, but they've got to get into the nucleus where all the DNA is in order to be turned on. The other thing is we know that AAV vectors inside muscle cells often combine with each other and form these very large circles of where you may have 20 different copies of AAV all joined into a giant circle like a Ferris wheel. There's some evidence that perhaps in those circles many of those AAVs are shut off and they're not able to make a protein. So we've actually started a series of experiments in our laboratory to try to address all these issues and try to come up with a reason why some AAVs are active and some are not. But I think you're exactly right. The amount of AAV that's getting into muscle tissue is at the level where it should be completely therapeutic, but something seems to be shutting down some of those vectors. And if we could figure that out, you could turn those on, and we might get a real boost in how much dystrophin is being made.
Can I just ask one follow-up for that? Do you envision a world where boys may indeed have viral genomes? And by viral genomes, we really mean they have the microdystrophin gene in the cassette. There's no virus there anymore, right?
Right, exactly.
So
Yep.
the DNA that's being put in is delivered by the virus, but the genome itself is the transgene. And so that transgene lives a long, long time now, at least 7 years out. Do you imagine that there's some of those not being expressed 7 years out that could be reactivated? So just ask you a provocative question.
But potentially, yes. Yeah, I think it could. And yeah, you bring up a good point, and which I usually try to make. I often forget. These AAV vectors, they're derived from a virus. They're not viruses. No viruses are entering the body. What ends up in the muscle is a pure piece of DNA with nothing else. And so, from a safety point of view, it's an important thing to keep in mind. Presumably, again, I think two things are happening. One, these dystrophin genes are either not getting to the right place inside a muscle cell to become activated, or they're being shut off for some reason. And in either case, you can imagine additional interventions that would drive those into an active state, which could bring up the level of dystrophin production down the road. And I think that's an important thing that people should be looking at.
And then I want to get to these last two questions before we move on to our next session. So we'll start here in the middle and then off to my right. Go ahead.
Hi, this is a question for Kevin. You talk about the exclusion criteria in the SOLID clinical trial, and you said it's for deletions 1 to 8, I think. What about the duplications? Like, no one's talks about that. So my son's got a duplication of 3 and 4. Would he be excluded from that clinical trial?
No, actually, duplications are not. So the idea is that deletion mutations are mutations that or mutations that interrupt translation before that point. It means there's no possibility of a protein ever being expressed natively with those portions. If you've deleted 3 and 4, you're putting back in a transgene that encodes 3 and 4 that the body has never seen. In a duplication sense, 3 and 4 has already been seen there as well. So duplication mutations are not excluded from the study.
Okay, and the second part of my question was, some of them require less than 5% dystrophin to participate in the clinical trial. So my son's had a biopsy and it shows he's got 30% dystrophin, but they can't tell me how much is actually working, if any. So how do you test for that?
So maybe we're happy to chat afterwards, because I was going to ask separately. In our databases, duplications of 3 to 4 are typically much more on the Becker side than the Duchenne side. And we've also biopsied boys who have significant amounts of dystrophin with that specific mutation. So there's two reasons to consider the idea of not participating in the trial. One is the point of view of the parent. From the way I view it, the point of view of the parent, not exposing your child to the significant risk of a trial if in fact he's already making a lot of dystrophin. The other side is the point of view of the investigators ourselves, to say a mutation that is always associated with a really milder version of the disease will complicate interpretation of whether something works or not, right? So even though he would not be excluded based on his mutation, a duplication in that region, I would counsel you that there's reasons to consider he should not be, would not be a good candidate for the trial today, right? I would, but 3 to 4, I'm happy to talk to you about it. We know we've collected actually a group of patients with that specific mutation we can make some comparisons to that might influence your decision about it.
Thank you.
And then final question.
Yes. Good afternoon. I'd like to preface this by acknowledging that it has been briefly touched upon, but my intent is to dig a bit deeper. So my two-part question specifically pertains to our boys who have developed these antibodies, rendering them ineligible obviously for safety reasons. So firstly, what collaborative efforts are currently underway to address this significant barrier? Secondly, in addition, what specific roles or initiatives is your team directly leading in this area?
I can talk briefly a little bit about what PPMD is doing. So, I mean, this is something that, from our end, we do know is a major issue, and that we want to ensure that patients that have pre-existing antibodies, that is not a reason why they are excluded from potentially receiving gene therapy. So we continue to look for research that we can fund to that end. We are doing a collaborative grant with the Muscular Dystrophy Association and Cure Duchenne to fund some work out of Dr. Barry Burns' lab at the University of Florida to look at repurposing a drug that's used in another indication that we hope can maybe bring down antibody titers for patients that are probably closer to sometimes what the cutoffs are. It's going to vary, I think, as we look at what patients' antibody titers are, depending on how high they are, whether some of these drugs might work. But from an organization standpoint, that is something that we are keenly looking into and trying to invest in. And then I don't know, in your labs, if you've been looking at anything in particular.
Well, I wouldn't talk about what we're doing. Our lab's not working on it, but I think it's worth knowing what's going on out there, right? So there's kind of three different approaches. One is currently in clinical trial, actually, with plasmapheresis, which is a method of rinsing out's not the term, but filtering out immunoglobulins out of the bloodstream. It's a common procedure that's used for autoimmune disorders where you want to get rid of antibodies. There's a clinical trial. Sarepta has a trial underway to try to do plasmapheresis. Plasmapheresis is rough. It's not without risk. You have to put in lines into the body and filter blood out and return it, for example, and you go through multiple rounds of this to try to get the antibody level low enough to go ahead and treat. So that's one approach. A second approach is to find other ways to knock down antibody responses. So one of these would include certain peptidases, the peptide degrading molecules that multiple groups have looked at, including Barry Burns and others, to think about, can you give these to knock out IgG, the class of antibodies that we worry about? Can you give these as a predecessor can you do that safely in order to knock down AAV antibodies and to treat a patient? Or another route is to give rituximab before, to try to knock down all the IgG producing cells in a way you could do it. So people are looking very actively about this in experimental animal model ways. I'll also add, we've not really touched on non-viral gene delivery. So there are really interesting things coming up in non-viral approaches in which your antibody status will be irrelevant to the delivery for this. And I don't know that those are really covered by any of the talks here, but there are serious groups, serious companies looking at this as the next generation for us to move forward.
Awesome. Thank you so much.
Well, we're going to, I think, wrap here, and I'd like to thank all of our speakers and our panelists for this great conversation.